Good morning. My name is Noah Trebesch, and today I'm going to be talking about software called Xsys Builder, designed to turn meshes derived from experimental sources into fully atomistic membrane models that can be simulated using molecular dynamics, or MD. Specifically, I'm going to be talking about modifications to Xsys Builder that we've made over the past year to enable it to incorporate membrane proteins into the models that it produces. Our motivation for developing this software is the desire to simulate real cellular membranes such as these. These membranes have three characteristics that we care about. They're immensely large from the perspective of atomistic modeling and simulation. They have very complex lipid and protein compositions, and they also have very complex geometries. There's no modeling software out there currently that, other than Xsys Builder, that's able to handle all three of these features of these types of membrane systems. In developing Xsys Builder, we've used a very small piece of the endoplasmic reticulum called the Terasaki Ramp as a representative cellular membrane. It has a complex helicoidal structure that was determined using electron microscopy in 2013. We've also incorporated a coarse approximation to the lipid composition of the ER into this model, and I'll be using this coloring scheme throughout the rest of the presentation. Going after MD simulations of the Terasaki Ramp or other structures like it is very ambitious. However, at the NIH Center for Macromolecular Modeling and Bioinformatics, we have a long history of pushing the boundaries of what's possible in terms of scale for MD simulations, and so we feel confident that we are well equipped to address this challenge. I don't think most people in this audience needs an introduction to MD, so I won't dwell on this, but briefly, MD works by defining a potential energy function called a force field. There are a variety of terms in the force field that hope to capture the atomic interactions. There are terms for this to describe bonds, angles between bonds, dihedrals, etc. And using this potential energy function, as well as the initial position of the atoms in your system, you can iteratively integrate Newton's equations of motion and throughout time obtain, for all the atoms in your system, updated accelerations, velocities, and positions for the atoms. Using the changing position of the atoms over time gives you atomic dynamics, which collectively give rise to molecular dynamics. And we use this tool to try to determine the structural basis for the function of biological systems. Okay, so Terasaki Ramp is very large. It makes it impractical to use it to develop and test new modeling steps in Xsys Builder. And so we use this much smaller synthetic system. It still has complex curvature in order to test new modeling steps. And so I'll be using this system in a movie to highlight the key modeling steps involved in Xsys Builder. So the first step is to obtain a mesh from experimental sources that represents your outer leaflet. You need to use this to generate a mesh representing your inner leaflet, which you can do first by measuring the triangle normal vectors, which can be used to measure the vertex normal vectors, which can then be used to place the inner leaflet mesh vertices one bilayer thickness away from the outer leaflet mesh vertices. If you repeat this for all the vertices in your system, you obtain an inner leaflet mesh. These meshes must then be converted to grid-based potentials, which are used in the next step of optimizing the initial placement of the lipids in the system. This is done by representing the lipids as particles, simulating the particles while attracted to the grid-based potentials. Yes, and then after this simulation is done, you need to replace the lipids with atomistic particles, which can be done first by selecting a particle, selecting its nearest neighboring particles, fitting a plane to the selected particle, and then assigning the plane normal vector to the originally selected particle. The particle can then be replaced with an atomistic lipid from a library of conformations, and this can be repeated for all the particles in the system. At this point, you have an atomistic representation of your structure, but it's not yet ready for simulation. You first need to detect complex lipid clashes, such as ring piercings, and fix them. And Xsys Builder has an automated way of doing just this. So once you fix these clashes, you're ready to solvate your system and address the challenges associated with solvation, and then finally you can run your simulation. Okay. So after applying all these steps to the synthetic system, we were able to run an unrestrained equilibration of this synthetic system, and it's shown here. The top is a side view of the synthetic system, and then these three panels show our cross sections at 3 different locations throughout the system. And then these blue surfaces represent the boundary of water. And so there's no water in the inside of the membrane, but there is water here on the inside of the structure and on the outside of the structure. So what you're seeing here is that the overall shape of the system, which is composed of about 12 million atoms, this is a 180 nanosecond simulation. Overall, the shape of the system is relaxing, which is expected because there aren't any proteins or anything else there to maintain the curvature of the system. But if you look at the cross sections of the membrane, overall their integrity is maintained, which was not the case for earlier versions of this simulation. And we're willing to say that we're fairly happy with the results with the model that Xsys Builder was able to produce. And so with this result, we moved on to trying to incorporate membrane proteins. And so, excuse me, the first step in incorporating membrane proteins is to generate a packing of the structure. To do this, we use software called Cellpack. Cellpack represents proteins and other elements in the system as meshes, and it's able to produce very complex and dense packings of these meshes into different compartments, and also do so at a very large scale, as demonstrated in this figure from the Cellpack paper. So we used it by use the synthetic system as the shape of the structure, as the representation of the membrane, and chose 2 arbitrary proteins to pack into the system. We ended up with a packing of 25 copies of each protein from Cellpack. There are several modifications to the methodology of Xsys Builder that we have planned, but they're not trivial, and they'll take a little bit of time to implement. And also, from the experience of building the lipid-only system, we believe that quickly generating a coarse model of the synthetic system with proteins incorporated in it, using a quick and dirty modeling approach would be very advantageous. It would help inform our planned modifications, and so we proceeded doing that. So basically what we did is we took the packing that Cellpack gave us, we converted the mesh-based representations of the proteins into atomistic representations, and we overlaid that with the lipid-only structure of the synthetic system that I showed a simulation of just a few slides ago. With this overlay done, we deleted all of the lipids that were overlapping with the protein to eliminate all the clashes in the system. And we note that this is not an acceptable protocol for the final modeling or final approach for Xsys Builder, because it deletes too many lipids. You can sort of see there's a ring or a halo of space between the protein and the membrane environment or lipid environment. And it also is deleting lipids blindly, and so you're not maintaining your lipid composition using this approach. Nevertheless, we proceeded with a restrained equilibration of the system, holding the shape of the membrane very rigidly in place, as well as preventing any water from passing across the membrane, and holding the backbone of the protein in place. These blue surfaces you're seeing are indicating that there are large vacuum bubbles that are forming inside and outside of the system, which is an expected result. This is the same thing that we get when we do the lipid-only system. So we have a method to, we understand why that's happening, and we also have a method to correct it. So corrected it, and then we're able to run a unrestrained equilibration of the system. And immediately, upon running this simulation. So this is with the proteins. It's about 25 million atoms. Ran the simulation for about 10 nanoseconds. Immediately, we were able to see that there were a number of issues or modifications to the modeling protocol that we would need to make. Noticeably, there's sort of a hole that's forming in the membrane at the center of this packing of 5 different copies of the red protein, V, C, and D. And then these blue surfaces are at these different points are showing that the inner and outer leaflet are unzipping from one another, and large vacuum bubbles are forming inside in between the leaflets. So this hole is forming because the proteins are just packed too close together. There isn't enough space for lipids to be placed in between these proteins in the initial model. And also, we encountered an issue where the polypeptide chains of 2 different proteins actually were interlinked based on their placement by Cellpack. And so by default, Cellpack places proteins randomly across the surface of your membrane structure. We will modify this so that there's a little bit more space between the proteins. And then there are 2 reasons that this unzipping between the inner and outer leaflets is occurring. The first is that the membrane proteins were not inserted deeply enough into the membrane. We use Cellpack to insert them such that the center of the proteins were placed at the correct theoretical level within the membrane. But this, of course, hindsight is 20/20. This, of course, does not work for where the proteins actually meet the membrane, and so you have a large mismatch between the hydrophobic ring that surrounds all membrane proteins and where the membrane is actually being placed. And so what happens during the simulation is that the lower leaflet, they're placed up against hydrophilic residues. They don't like that, and so they sort of crawl over the bottom of the protein and then link back together, and that zips them away from the outer leaflet and decreases the overall surface area of the inside surface of the membrane. The second factor has to do with the outer leaflet. And so I mentioned that we were mostly happy with the simulation of the lipid-only system. We weren't entirely happy with it because it turns out that it's overfilled. During the simulation, we ended up observing these Y junctions like this with portions of the outer leaflet forming their own separate bilayer jutting off from the main structure. And these 2 results led us to reevaluate the way in which we were calculating the number of lipids to fill the inner and outer leaflets. And so we redid the simulation with lipids only using a new method. And this method does not have these same Y junctions, protrusions. The overall, much more happy with the results of this simulation. So we also have a correction for that problem. Okay, so going forward, the path is pretty clear. We will implement the new modeling steps that we have in mind for xMAS Builder and also take into account the results from this simulation. Then work on building a new model and run that model, and then eventually start applying it to larger systems once we have everything worked out. With that, I'd like to acknowledge my advisor, Ahmad Taj-Khorshid, more broadly the members of the Taj-Khorshid group, and the NIH Center for Macromolecular Modeling and Bioinformatics. I'd like to acknowledge funding sources and computer time that allowed me to complete this work, especially Blue Waters, where the majority of the simulations you saw were run. So thanks.